on the call, and I'll introduce our speaker today in a couple of minutes. And just a reminder, I mean, we do record this presentation. In fact, for our previous guest lectures and webinar, and it's also on the YouTube channel. I just put it on the chat. So if you want to check out previous speakers from the past, you're welcome to follow that link and check out the other speakers in previous weeks.
And just to double check, we end at 3 p.m., right?
Right.
As at 3 p.m. my time.
Yeah, 1 hour. Yeah.
1 hour. Yeah.
Okay, so let's get started. Hello, everyone. This is Jiming Sun. I'm a professor at the Computer Science Department, and also a joint appointment at the Carleton, Illinois College of Medicine, and also associate faculty member for the neuroscience program at UIUC. It's my great honor today to welcome one of us coming back, actually, Dr. Faraz Faghri, now as the lead scientist at the Center for Alzheimer Disease and Related Dementias, CARD, at the NIH, and also partner at Data Technica International. He earned his bachelor degree in applied mathematics and master in industry engineering and Ph.D. in computer science at UIUC. And his thesis focused on learning long, healthy aging and involving developing machine learning model for aging-related disorders. And he also completed a fellowship in technology-based healthcare at Mayo Clinic. His work has received numerous academic awards and has been transferred to tech and biotech industry. And he also strongly supports open science, technology, democratization, diversity and inclusion. And in particular, he contributed to the creation of a lot of exciting projects I hope he will talk about today, including GenoML, CRISPRbrain, and CRISPRlipid. And without further ado, let's welcome Dr. Faraz Faghri.
Thank you very much for the great introduction, Jiming. It's exciting to give the talk at UIUC. So this talk is going to be a little bit of overview about what we're doing at CARD, and then jumping to some of our priorities, some of our projects that we're working on. And I really like to ask that make it interactive, and you can just ask questions. I don't know, however, how the format is for this particular presentation, but you can post your questions, and I'm sure Jimmy would bring it up. But let's start with what are we doing at NIH and at CARD, and then better realizing why do we actually need some of the computer science projects that we're doing to be done at the NIH. So CARD, or Center for Alzheimer's Related Dementia, is one of the recently established centers at the NIH. And it focuses on specific Alzheimer's disease and related dementias. And related dementias would include a spectrum of neurodegenerative disorders. So the projects that we tend to do at NIH are are relatively high risks, high risk projects that is much more costly or much more or too large for an academic project. So as many of you may know, NIH would give these grants to the universities, but it also keeps some of the budget that Congress allocates to NIH and spends it on projects internally. And those projects, again, are high-risk projects. They are projects that require a lot of collaboration. They require a lot of flexibility in terms of technologies that we use. Projects that would require huge infrastructure, whether it is human infrastructure or data infrastructure or compute infrastructure. And projects that require a level of coordination that is not available to many academic institutions. And specifically CARD Center, where we are at, we focus on a new way of looking at Alzheimer's disease and related disorders by removing a lot of boundaries that has been put in between disorders. So we like to think about neurodegenerative disorder as a whole spectrum and not looking at them as individual silos. So with that, this slide, actually a lot of slides that we go through are heavy in terms of biology and clinical practice. And I think that's probably one of the biggest things that I missed when I was doing my grad studies at university, where we would focus on problems mostly from a science standpoint and not taking into account the applications of them. Whereas here at NIH, we actually look at the problem and we say, all right, we have this clinical biological problem. How do we address it? Whether, and maybe the solution is just a simple statistical solution, or maybe a very fancy machine learning method. So it's a little bit different than the way that we usually operate in the computer science department. We would like to go with the fanciest way of a fancy solution. Here, we want to just solve the problem. And in that sense, the problems that we are dealing with are as follows. We have 4 scientific theme type thrusts in our work. We like to work on molecular pathogenesis of that are related to genetic and the genomics data. We like to work on disease subtyping, prediction and risk and progression of disorders. We work on de-risking of therapeutic targets. And by that, it means if you're working on the therapeutics, medication, or some other method like gene editing, what is our target and how do we minimize the risk of going after a target? And it is very important to realize why are we talking a lot about risk in this presentation when I go to the next part, telling you a little bit more about disorders, but the de-risking part is important. And then focusing on development of application of precision therapeutics. And we have structural priorities. We need to make sure that we not only have both diversity in terms of research and researchers, but also the way that these therapeutics are targeted for different communities. Making sure the data that we generate or we manage is publicly shared with the community. Making sure everything that we do in terms of code or practice is transparent and reproducible. Bringing collaboration from different groups and having foundational systems that anybody else can use and enable their research. All right, so that was a little bit of what we do at CARD and how things are different at the NIH. I'd like to now jump into the scientific overview, and it's a little bit of how you need to think about these problems, the framework that is important for you to think about these problems. So human aging is a very interesting process. We go from infancy and we go through multiple different stages in our life. And this aging is complex, and each person has their own pathway. So even though we might start similar to each other, as we age, our trajectories are going to be very different. And that makes some of the research that we do and the work that we do harder and harder. And one of the important questions that we have in our work is, could we use machine learning to construct this aging model? And this aging model that we work on is focused on adulthood, post-adulthood. So we don't work on pediatric research. We really work on more after reached adolescence and then work toward the aging part, which means the growth has finished in the clinical setting. It means that there is no more growth in a sense. Now we're just, some sense, it is downhill in terms of cell loss. So what are some of the challenges that we have in this type of research that we do from the computer science standpoint? One is that we have lack of reliable labels. So it is aging, and average age used to be around 40 years, but now our average age is around 72 years. So it means that we are trying to model something that we haven't seen yet. And if we have seen it, we don't have proper labels for it. So a lot of times people might get diagnosed with a disease, let's say Parkinson, and years later, we might realize that there was a misdiagnosis because we didn't understand how the disease, and it happens a lot in our work that we actually don't have quite reliable labels in our data sets. So even if we have a label of Alzheimer, it doesn't necessarily mean it was Alzheimer. Maybe it was cardiovascular disorder that caused dementia. Maybe it was frontotemporal dementia or something else. The other part is utilization of short-term and long-term longitudinal data. We have that a lot, as in we're talking about decades data. We're talking about data that we need to analyze data sets that has been collected over multiple decades. Some of them might be shorter, some of them might be longer. So the longitudinal aspect is very important for our analysis. Integration of multimodal data. The other aspect is, in order to better understand the aging, we need to focus on multiple different data sets, imaging, genomic, proteomics, different types of brain imaging. So that's another challenge that we have. How do you bring in all these different modalities? And at the end of the day, we need to interpret the results. What it means is that we can't just say, oh, our model works great with 98% accuracy. You need to show why your model is doing what it's doing. You need to show the situation that your model is not going to perform well. And you need to explain to the clinician how they need to communicate the results to the patient if they are using that model for patient counseling. Now, these are all engineering challenges. We also have clinical challenges, and some of them is we need to replicate our results in an external data set. For us to actually use a model, one of the biggest steps is, all right, you have this model, it was trained on this cohort. Let's say this data was from US, you have a model for US, but you need to replicate it in a data set that comes from, let's say, Germany, to see how robust the data is, to see how generalizable the data is. So we don't usually get to the finish line until we have that replication in an external data set, which adds another challenge. Most often we don't have an external data set. So how do we find that external data set to use? We need to make sure the model is usable. So we can't just say, oh, this is great AUC and here are great parameters. You need to figure out why is it usable. So it actually involves a lot of human-computer interaction to communicate with the users, to communicate with the physicians, with patients, with biologists. How are they using the model? Does it make any sense to them? Is it what they need, or is it what is helpful to them? So that's another aspect. And at the end of the day, we need to show that it's providing tangible improvement in decision-making and clinical trial design. So a lot of very, very, very practical and applied research here. Not much theory in that sense. All right. So our data-driven strategy or work has 3 phases. In the first phase, we collect data, which is a lot of different modalities: brain imaging, genetic, clinical data, environmental data, environmental exposure, virus, toxicity, all these things we collect. And it might be from many different sources: public, private, semi-public. Then we do some magic that you are all experts in. And ultimately, we like to provide 3 values in terms of public health values. The descriptive values are: we like to describe types and subtypes of heterogeneity and interplay between disorders, identify many-to-many genotypic and phenotypic relationships. It's sort of this phase is sort of unsupervised learning in many ways, that we like to extract new patterns that we don't have labels for. The second part is we like to provide predictive value, predicting disorder susceptibility, diagnosis, prognosis, when the disease is going to, when the onset is going to start. And those are prediction parts, a lot of supervised learning in that sense. And the prescriptive part is that, all right, knowing where a patient is going to be, where their prognosis is going to be, what's their diagnosis, could you tell me what treatment and intervention could I introduce in terms of managing the disorder, preventing it, or treating it? All right, so with that framework in mind, and I'd really like to emphasize on all these elements, because you might have been exposed to a lot of research papers in different areas, for instance, cancer, and when say, oh, this problem would be easy to solve if it was cancer, and that's true. In neurodegenerative disorders, the challenge that we have is by the time a person is diagnosed with a disorder, let's say Alzheimer, it means that the disorder had already started 2-3 years before that. And it takes a few years before a patient becomes symptomatic. And then the prognosis might be between 5 years to 12 years or even longer. So if you put it in the context of data collection, it means that you have to spend a huge amount of effort to collect data and analyze these data sets. All right. So one of the biggest targets that we have, important goals that we have, is moving toward an open source precision intervention. The translation is, we like to have a clinical trial, a first open source clinical trial for these disorders. And our pipeline for getting to the clinical trial is as follows. We start with modifiable risk factors. We like to identify the risk factors that are already identified, and then see how we can change them. And we have some projects in that area, and it involves mining a lot of different databases. We work on identifying the target. Many targets fail. And one recent study showed that if you have a target that's genetic based, it has higher chance of success. So a lot of work that we do is on the genetic side to identify targets that can be checked, how they're involved with the therapeutics to minimize the failure. And we do due diligence, figuring out how the risk, taking care of the risk. And again, I emphasize on the risk. The risk is important because if you have a treatment, it will take at least 5 years for the clinical trial to show how the results are the results promising or not? Is that therapeutic promising or not? So you want to minimize your risk. You don't want to start testing something and then decade later, you realize that, oh, it was not a good target. So you do as much as you can to de-risk that target. And there are so many factors involved with risking. Not only it is the factor that how is it involved in biology, but also different population. Maybe the target that you're working on only works on 1% population. Should we pursue that or not? And we have some projects in this area. Precision clinical diagnosis. So now you have your target. Who should you test this target with? How do we test these in a clinical setting? And we have these machine learning models to better tell us what is the diagnosis or prognosis of a person, and whether we should work on, we should test that, and how would be the success rate of that therapeutic. And then the readouts. The readouts would be, all right, did it actually work or not? And we have projects in that area. And again, potentially like to get to the first open source clinical trial for these disorders. Now, I will jump into some of the projects that aligned with these steps in the pipeline. So I start with the modifiable risk factors and go on. I'll show you some of these projects. One of the interesting projects that was published recently, and it was led by Kristen Hampton, was based on modifiable risk viruses in neurodegenerative disorders. So recently, there was a paper, a very interesting paper, that they showed that Epstein-Barr virus is associated with multiple sclerosis. And one of the questions that we had is it just one disorder, or are we talking about more viruses and more disorders as well? And the great thing that they did, Kristen Hampton, was to go through two major biobanks, UK biobank, which is essentially the EHR in the UK, and FinGen, which is the similar biobank of clinical hospitals in Finland. And by mining these two biobanks, they realized that it is significant. So this is the chart, and the paper is now published in Neuron, and you can take a look at it. But we realized that there are multiple different viruses that are associated with different neurodegenerative disorders. So AD is Alzheimer, dementia, MS, Parkinson, and vascular dementia. So for instance, you have influenza that is associated with vascular dementia. And one of the hard part about this project, as I mentioned earlier, was the replication. So it was essentially this, we had a discovery cohort and then a replication cohort, so FinGen and UK Biobank. So quick summary of that work was that we have 22 replicated pairs of viral exposure related to neurodegenerative disorders. We're working on a further replication of it in a SAIL biobank, which is another biobank in Wales. The vast majority of these pairs include neurotropic viruses that cross the blood-brain barrier. And it's important because there has been a lot of hypothesis recently about what happens when we have certain viruses in our body and how they affect brain. COVID is one of the great examples, and we have, for instance, a project going on right now on COVID brains and association of that with neurodegenerative disorders. And we have many other disorders. And one thing that was interesting about this was that most of these virus exposures are preventable, and there are vaccines for them. And we can easily modify it in a sense that these are modifiable risks that can be done.
There's the question, maybe you can.
Sure. So the question is, are these viruses that patients were exposed to in the past? If so, how does one know that information? Are these some kind of traces of the virus still in the human body? Good question. So this was a biobank study, meaning that we didn't do any lab testing. This project was not a lab testing project, nothing in dish or in humans. So the way we knew that was that a patient in the UK biobank, you have the information about a patient going to hospital with an exposure to a virus. And usually that exposure must have been very critical, as in you don't go to hospital if you have just a cold or just a mild influenza. You go when it's something serious. So they have gone to the hospital, they've had that exposure, and they were diagnosed it. And then after that, they were, years later, they were diagnosed with a disease, with Alzheimer, for instance. And that's what we are looking at. All right, thanks for the question. So now in terms of due diligence, this one is more computer sciencey. So the earlier project that I explained, you could say it was a classic statistical project. And now you have these targets that you know, right? We have these certain genes that that some of our association studies have shown. I think I need to give an explanation of what is an association study first. But in genetic, what we have is we recruit a large cohort of individuals with a disease, let's say Alzheimer, and we recruit a large cohort of individuals without that disease. So that would be our control. And we look at the genetic data and we say, all right, in the Alzheimer cohort, these genetic mutations stand out. And these are possible targets associated with the disease, right? So we have these studies. And in this line that says input, that's a lot of that data coming in. So we have the SNPs per gene from GWAS. So GWAS would be the genome-wide association studies, the study that I just explained. And we have more information coming in. For instance, the burden statistical per gene. What is the likelihood of a gene associated with a disease? Then we have fine mapping per gene per GWAS. Again, that connects to the pathways of genes. Mendelian randomization, information about whole blood expression. Is that gene expressed in the blood? Is that gene expressed in a different organ or a tissue? So we have all this data associated with the gene information. And not only we have that for neurodegenerative disorders on the right, but also we have them for related disease data sets, including hypertension, diabetes, heart disorders. So these are all disorders associated with neurodegenerative disorders. And we get all this data, we add it with advanced data. So we have this CRISPR studies going on right now that we take this stem cell of people who have had a disease, let's say Alzheimer or Parkinson or other neurodegenerative disorders, and then we test different CRISPR gene editing methods in those. And we also collect data associated with that. If we changed a gene, did it make the neuron to live longer or no, it didn't? And then where it comes next is to the fun part, the network phase, the network analysis, that we have all this information brought into a network, a knowledge graph, and we'd like to use that knowledge graph to further do due diligence on the targets. Okay, and we have a prototype for that project. The aim of this prototype that I'm going to show you is the sort of the power of adding more information in a knowledge graph and how we're going to use that for our due diligence. So this project is called OmicsSynth, and I'd like you to pay attention to two numbers also on this slide, on this video. One is the number of nodes These are the genes in this network. And then the edges. The edges would be more information added to this graph. And here on this side, which says gene disease, I will keep indicating what data is being added to this network. So we start with no data, pretty much. We add some structural info. Nothing much. We just know that. And the big circles, AD, PD, ALS, are three most important neurodegenerative disorders: Alzheimer, Parkinson, and ALS. So nothing much. TYK2 is a gene. There are therapeutics going on, specifically with TYK2 for Alzheimer and inflammation, for MS and inflammation. GRN is an important gene associated with inflammation. LRRK2 associated with Parkinson. ApoE associated with Alzheimer. So now those are some of the important genes, and we like to see how they get placed in this network as we add more and more data to this graph. So now we are adding blood EQTL, substantia nigra EQTL. These are how a particular gene expressed is in part of body. Brain EQTL, brain EQTL. And as you can see, the network is getting closer and closer to islands, sort of SNVs, neurodegenerative disorders, deletion and insertions. Okay, now I zoom in this part, and then we have Alzheimer, Parkinson, ALS, and we have all these genes here. Okay, and you might have noticed we have at this point 346,000 edges. So you can think about it in the metrics part. Also, a lot of money spent on GPUs for this prototype, and also all the edges are removed in this for the visualization, and we only show them when we check a particular gene. So now I like to see an important targets. Where are they? So we have LRRK2, which is again an important gene associated with Parkinson. It's landing closer to Parkinson, but still it has some association with AD and ALS. We have ApoE, which is closer to AD, as it's more associated with Alzheimer. C9orf72 is one of the gene mutations highly associated with ALS. GRN is inflammation. So now that I have this graph, and this graph is showing what we also, our hypothesis is also showing for these neurodegenerative disorders, now I like to go the next step that we haven't pursued yet. The next step would be, all right, how are these genes, what are the pathways that they are associated? If GRN is having a change, how is it impacting other genes down the line in the pathway, or other biological elements in that area? So now we can do further work with this matrix. And that's one of the great, I would say, data sets and prototypes that we have right now created for this due diligence, and it's ready for more fancy graph analysis. All right, now let's go to the clinical diagnosis project. One of the projects that we have worked on, led by Mary and Mike in this, was to build models that would predict a person's susceptibility to Parkinson's disease, and whether they are going to get Parkinson's disease and when are they going to get it. And the data that in this study, the data is a combination of genetic data, transcriptomic data, and clinical data. And it goes through a huge amount of quality control and filtering, because that's one of the biggest challenges with genetic data. you need to make sure that your data is high quality for the models, for the machine learning models. I would say when it gets to the machine learning model training, the project is 80% finished, because a lot of work is spent on the quality control and gathering the data and cleaning the data. So this project produced a model with relatively high AUC, and 70% AUC of predicting someone would get Parkinson's disease only with genetic data. 80 is a 7% when we add the clinical data. And when we add the combined of everything, it goes up to 89%, 89.72%, which is significant for clinical setting. That's a significant number to know. And we also made that package publicly available. It's GenoML. So the whole process of giving the input data, phenotypic data, genotypic data, all of that, and making a pipeline of munging harmonization, all the quality control, training the model, validating the model, and then generating the outputs that are accessible for, are now accessible for other researchers. And we have many people who are using it for clinical trial and in-house. So we're very happy with the success of this project and this particular project, GenoML. Similar to that, so that was for Parkinson. This one is for ALS. This one was led by Anand, who is now doing his Ph.D. at UUC. It was working on subtyping ALS and building a model that would predict a person's subtype in ALS, again, using the machine learning models. And one of the great things about this project was that this was, again, reproduced. So it was trained on a cohort in Italy, and it was validated in an external cohort from Germany. And I would say the hard part was done by the clinicians who collected the data over 2 decades. And these are the numbers for the AUCs, and 98 and 96 for replication validation. Another project related to Alzheimer subtyping. This one used a public, started with a public data set at the Alzheimer Disease Neuroimaging Initiative. And we are, the AUC on that project is around 90%. And these are all, again, multimodal data sets collected over a long period of time. This particular study was over 4 years. Another exciting project, and exciting in a sense that it is extremely hard. And it is also being led by Anand at UUC, is a project on imaging and genetic data harmonization, imaging and genetic. And this project is extremely hard because of the way it started. So our goal was to harmonize every brain imaging data that's out there in one place. And that includes all these public data sets like ATNI, PPMI, NIAID, and also UK Biobank, process them and bring them in one place for the machine learning models. And to give you a sense of the challenge, here is the input. Each MRI T1 image is about 25 gigabyte. Okay, so if you want to put it in the context of imaging projects that is typically done in the machine learning community, you have smaller images, but you have a larger number in terms of sample size. Whereas in our case, we have huge images, so 25 gigabyte is each image, and the sample sizes would go maybe around a few thousand. So very different challenge in that sense. And for this problem, this was in collaboration with folks at In Vitro, who developed the package called AntX. We use this deep learning pipeline that takes the MRI T1 weighted image. It removes the brain from that image. So essentially extracting this skull area, and then taking it into the denoising pipeline, and then helps us extract about 400 features about this image. So these 400 features would be thickness of a region, volume of a region, the surface of a region. And these 400 features are essentially summarizing that MRI image, the 25 gigabyte image. And then we take that for our next steps. So at this point, we can actually have an end-to-end pipeline that we would take, like you do in a lot of image projects right now, that you would take all the images, fed into your network, and then go from there. Whereas in this project, we extract features from each image with a CNN-based model. Now, how do we use that? It's a very, very interesting project right now. So our motivation is that we can make these neurological disorder models from this data set, essentially showing, for instance, for tau pathology, tau is one of the proteins accumulated in the brain. Amyloid pathology is another protein. How and where do they start in the brain, and how the pathway is going as someone ages? So the T would be the time span. How does this protein accumulation starts, and how does it change in the brain? So, and when we look at the healthy brain and severe Alzheimer, you can actually see that you have atrophy in brain of these patients. Now, with this data, we like to construct this model of how this model is actually changing. And for this, when you have the 400 features, we use this XGBoost for making a model that does the prediction for each disorders, and then using the SHAP values to see which part of the brain was involved with that decision making. And the result of that is, for control and dementia, we can see as time passes, which part of the brain gets involved with that particular disorder, and how does it change. Now, having this model, in the current project that is being done, and that's for Parkinson, our current project is having this model that you have from these brain regions. Could we connect that to the genetic? So what is the underlying mechanisms associated, the genetic mechanisms associated with this movement? Which is an ongoing project, and we're hoping to have interesting results very soon for this one. Okay, so I thought it would be good to just summarize some of the projects up to this point, and tell you a little bit about some of the data sets that we are also generating. One of the biggest projects that is going on is related to mechanistic questions associated with the neurological disorders. So around 2009 to today, most of the effort in the community was spent on figuring out which genes have an effect on neurodegenerative disorders. That was the genome-wide association studies, GWAS, that we talked about. And there are so many different tools available for that. So GenoML that we talked about, or Terra platforms, Anvil, these are all platforms that have worked on this area. From 2020 to 2025, what we're going to work on and what the community is working on is toward answering these questions. Which cellular processes are affected? What loss of functional gain is associated with these genes? Which cell type are associated? How does this cellular environment protects this specific cell types? And a lot of these questions are being tested on stem cells, neurons that are in dish, and they are derived from stem cells of patients. And a lot of the tests are CRISPR tests. And one of our efforts is to make these data sets available to the public so they can also go and do their analysis as we test them. Because these are very expensive experiments, and we like to essentially do the heavy lifting on generating the data sets and experiments, and then make it available to others to work with. And from 2020 to 2025, another part is now testing it in the humans and see how the patient's lifetime is being affected by that. And that's another part. But the data set, part of the data set that we're developing, now we're putting in two platforms. One is called CRISPRbrain, and the other one is CRISPRlipid. So this is ORG. So CRISPRbrain.org and CRISPRlipid.org are two of the platforms that where we are making these data sets publicly available, and you can just go and see what's being generated in the community. And this is one of our fascinating projects done at NIH with our collaborators at UCSF and UC Berkeley. And in terms of people using the data, we are also looking at many people from many places in the world are actually using these data sets that we are putting out there. So that's our user map. There are other resources that are being generated in coordination with us, and one of them is the Global Parkinson Disease Genetic Program, GP2. Huge effort, huge project, and the goal of this project is to have 92 cohorts, 140,000 samples that are Parkinson, and one of the biggest effort is around diversity in these data sets. So 2,500 samples from Nigeria and Egypt, 500 from Latin America, more than 1,000 from Asia. So to get a better sense of the diversity of these disorders as well. And this data is being collected and analyzed, and right now a good portion of the data is publicly available that can be accessed through Terra, which is Terra Bio. If you go to Terra, if you create an account, and access the data through the GP2 access line. Another one is the iPSC Indie initiative. It is related to creating stem cells and then having different cell lines associated with different gene mutations. And that data set, again, huge amount of effort. And that data set is already public and available through ADWB website, ADWB platform, which is on top of Azure. And people can go and use the data over there. Both Terra and ADWB data is there, and you can just go and analyze data on those platforms. Terra is on top of Google Cloud, ADWB is on top of Azure. All right, with that, I would like to have some time for questions. So thanks for having me, and I'm looking forward to questions.
Thank you. Thank you for us. This is fascinating, especially a lot of useful resources and pointers. I think we'll have our students can learn a lot from this talk. And for audience on the call, if you have a question, you can either just unmute yourself, ask directly, or put it in the chat. And so while we're waiting for those questions to come in, and typically we have some general questions that we run for the guest lectures. I mean, since we have a lot of grad students, right? You were a computer science grad students yourself, quite a few years back. So can you offer some advice for our students, like what skills they should gain in their graduate study? Maybe this sounds like a general thing, but I mean, from your talk, I gather you must have gained a lot of biology domain expertise. I don't know whether that happens during your PhD or after. So is that something important? And what other skills do you suggest our students to pay attention to when they are going through the graduate study?
That's a very good question. So I would say a lot of, I know biology from YouTube. I have not taken any courses about biology or medicine. So pretty much everything that I come across with is searching it on YouTube and finding a video that explains it better. And sometimes it's hard because a lot of the things we do, there might not be a video for it yet. So you need to go and talk to people so they would explain it to you. I think that's the biggest skill. The skill would be finding people who are willing to teach you or to answer your questions in different areas. And I was lucky to find those people and being able to ask them the questions about different aspects of genetic or biology or medicine. I just realized I did take the bioinformatics course in grad school, so that's probably the closest biology course I took. But that's the biggest one, and it keeps going. So a lot of the biology work that we are working on right now, they are not in the textbook yet. So they are very advanced, they're cutting edge. So you need to, and we are dealing with those data sets and they are being generated and we have no idea if the data is good or bad. So we are taking, for instance, these images of cells and looking at cells. Is the biology good? Is the protocol good or not? And you don't know what the biologists are talking about unless you know the jargon and go and read about them. So that's really the toughest part. Another opportunity at UIUC that was very helpful to me was, for instance, the IGB, Institute for Genomic Biology, and some of the seminars they had on campus. So always going to seminars from the areas that you're interested in. Maybe you're interested in health, maybe you're interested in astronomy, but going to those seminars and talking to people and learning from them. So being curious at the end of the day, and also coming out of your comfort zone. So we tend to be very comfortable with some algorithm that we really know well, or some data set that we really know well. But the reality is, especially in biology and health, everything we're working on going to be obsolete in 4 or 5 years. All these data modalities that I talked about, 4 or 5 years from now, we have better technology to collect the data. It's actually interesting because we actually ignore a lot of genetic data that was collected 10 years ago, because we realized that the error rate in them is too high and they're too noisy, so let's just forget about them. And then you have to read and understand what is the new technology that's coming and what is the new way that you need to learn. So if you're comfortable in one area, you would be obsolete in 4 or 5 years. So you need to keep learning about, if it is healthcare, different data collection methods, different modalities. If you are in computer science, new methods that you need to borrow from the computer science and take it to your area. That's the sad news, probably, that yeah, if you're learning something today, you know that it's going to be obsolete. But at the same time, it's motivating because you realize that creativity becomes an important factor here, to not only learn new things and new techniques, but also matching them together. And I think that's another, that's the biggest thing that we have. When we work with some of the collaborators or the new students, a lot of time have no idea about the new algorithm they're going to use. So a lot of time they teach me about the new algorithm and the new technique that they are using, which is great because then not only you learn something, but also you realize that it is a teamwork and a collaboration work to find solutions for problems. Any other solutions, any other advice based on the time that I had? Talk to as many people as you can in your cohort and with your peers. You might realize that some of the people in your class would end up being your friends and might be interested in working on projects 10 years from now. So keep those connections. They're going to be very helpful in the future. Yeah.
Great advice. Like watching YouTube videos, attend seminars, make friends, and make connection, keep learning. So I think we have Benjamin has you're here, has maybe a more specific question. Benjamin, you want to elaborate or what you...
So the question is GenoML is using about 11 machine learning algorithms available in the scikit-learn to handle AutoML. Since this was the open source package used for multimodality Parkinson's purpose, do you expect to use the same set of learning strategy for the global Parkinson investigation? The answer, I answered in two ways. If we hadn't done that, okay, and not that we have done that, okay, because your question is, do you expect it's going to be? We didn't expect it to work. We didn't expect it as in, we come into a project with an open mind and eyes open and say, all right, we don't know what algorithm is going to be useful, so let's try everything and test everything. And all the, for instance, include the neural network ones, everything that has people that people have been talking about, and see if they are going to work better. So that was the expectation, that we come in open mind project, open to anything. And we have done that project, and the result is the boosting methods, those 11 algorithms still work better, and we just test them depending on and we sort of let them race and see which one is giving a better performance. And it turned out that it's still the same 11 algorithms.
Okay, cool. I think we're running out of time, but I think the video will be available online later, maybe next week. And let's thanks our speaker, Faraz Faghri, one more time. Thank you for this wonderful talk. And thanks for joining, everyone.
Of course, it was delightful. It was a delight being here. Have a great week.
Thank you.
Bye, everyone.